A plant and a fungus do not share a language. They diverged more than 1 billion years ago. And yet, right now, somewhere out in a field, a plant is packaging a tiny bubble of RNA, making it across the root boundary into a fungal cell, and the fungus is reading it and following those instructions. Sometimes to its own harm. We built our large language models last decade. Biology built the first one 4 billion years ago. I want to start with an experiment that I still find a little hard to believe. A researcher named Hailey Jin at UC Riverside was studying how plants defend themselves against fungal infections. She noticed something strange. The plant was not just putting up walls, it was sending messages. Tiny spherical packages called extracellular vesicles. Think of them as little lipid envelopes. Were being exported from plant cells and absorbed by the invading fungus. Inside those envelopes was RNA, not random RNA, curated RNA, specific sequences. And here's the part that stopped her lab cold. The fungus was reading the sequences. Its ribosomes were building to the plant's RNA, following the instructions, and in some cases, translating them into proteins that hurt the fungus itself. Jin calls these pseudovirus packets. The plant is essentially putting together a small, believable-looking piece of code, handing it to an enemy that has never seen it before, and the enemy's own internal machinery compiles and runs this code. Jin's own line on why this strategy is so devastating is the part that I keep coming back to. One mRNA can be translated into many copies of proteins. It's much more effective than transporting the protein itself. Sit with that for a second. The plant doesn't have to ship a weapon. The plant ships the blueprint. The enemy reads the blueprint with its own machinery and builds the weapon on its own dime, in its own cells, against its own interests. Now let's pause on that. A plant and a fungus last shared a common ancestor something like 1.5 billion years ago. They should have nothing in common, and yet a sequence of RNA letters written by one of them is not just readable by the other, it's executable by the other. The message lands, the machine compiles it, and behaviors change. A few years later, Susan Erdman showed that archaea, those single-celled organisms that live in hot springs and salt lakes, also fill extracurricular vesicles with curated RNA and toss them out into the environment. Amy Buck at the University of Edinburgh looked across every kingdom of life she could get a sample from and found the same pattern. Bacteria do it, animals do it, plants do it, fungi do it. Every domain of life is sending each other RNA postcards, and every domain of life can read them. The first time I read about this, my honest reaction was, how's that physically possible? How can a message written by a plant cell be meaningful to a fungal cell that has no idea that the plant exists? The answer is the part I find quite astonishing. It works because the machinery that reads RNA is older than the kingdoms of life themselves. The ribosome, the protein assembling machine, is structurally almost identical across every living thing. The ribosome in your liver is recognizably the same device as the ribosome inside a bacterium in your gut and the ribosome inside an archaeon in a hydrothermal vent. They all read RNA in roughly the same way. They all use the same 3-letter at a time code. They all look at the same table of 20 amino acids. The translation machinery is a universal interpreter. It was fixed, more or less, before anything we would recognize as a cell within a nucleus even existed. That means that RNA has the property most languages don't. It has a universal reader. Not approximately universal, universal. Anything alive that still descends from the last common ancestor, which is to say, everything alive, carries around a compatible runtime. Drop a piece of RNA with a start codon into any cell on Earth, and there is a decent chance something will try to translate it. Amy Buck puts it in a line that I keep coming back to. She says, RNA already has meaning in every cell. It's a pretty simple code. And what she's pointing at is that RNA is not like a book, which needs a shared culture and shared language to mean anything. RNA is like a protocol. The format itself is enough. The cell on the receiving end does not need to know who sent it or why. The cell just has a ribosome, and the ribosome just runs the code. And this, for reasons I want to spend the rest of this video on, is structurally the same situation we find ourselves in with embeddings and large language models. Let me take a sharp turn into the computer science, because this is where the parallel gets really beautiful. When a modern language model reads a word, it does not store the word as a word. It stores it as a vector, a long list of numbers. 200 numbers, 500, a couple thousand depending on the model. That vector is called an embedding. And the trick of the embedding is that the numbers have no intrinsic meaning. You cannot look at the numbers and decode what the word is. What the numbers do have is a position. The embedding for the word queen sits in a very particular neighborhood of this multi-thousand dimensional space, close to king, close to monarch, but far from mushroom. There's a beautiful line from a Quanta article on embeddings that I keep stealing. It says embeddings are coordinates in interstellar space. Meaningless except for how close they happen to be to one another's known points. The meaning is not the point. The meaning is in the geometry, in the relational structure between the points. This is the move that made ChatGPT and Claude and every modern transformer possible. You stop trying to define what a word means, and you just train a huge model to notice which words appear next to which other words, across billions of examples. After enough training, the model has arranged every word, every concept, every fragment of meaning into a geometric shape where similar things sit near each other. Relationships become distances. Analogies become vectors. King minus man plus woman really does land near queen. And no human ever told the model that that's what a queen is. Now watch what happens when you put that next to RNA. An RNA codon, 3 letters, like A, U, G, has no intrinsic meaning either. There is nothing about the physical shape of AUG that means start a new protein here. The codon means start because the ribosome treats it as a start. It's just a point in a coordinate system. Its meaning is entirely relational. Change the interpreter and the meaning changes. Change the table that maps codons to amino acids, and AUG might mean something completely different. We know this because a handful of obscure microorganisms actually do a slightly different codon table. Same letters, same arrangement, different geometry of meaning. RNA, in other words, is an embedding, not metaphorically, structurally. It is a system where discrete symbols are arranged in a space whose geometry, the lookup table held inside the ribosome, defines what those symbols mean. Change the geometry, change the meaning. Keep the geometry stable across billions of years, and you have yourself a universal lingua franca. The transformer architecture did not invent embedding spaces. It rediscovered them. Evolution had already converged on the same solution 4 billion years earlier, because when you're an information processing system under selection pressure, this is the answer that keeps showing up. Here's the next puzzle, and it's the one I find philosophically the richest. If RNA is so easy to throw around, and if every cell can read it, why don't cells just dump their entire transcriptome, the whole library of RNA that they have at any given moment, out into the environment? Why package it carefully? Why select? Susan Erdman asked this question very bluntly. Her line, quote, "Why would you invest so much energy in throwing out random RNA in vesicles?" End quote. That's a microbiologist's way of saying selection wouldn't bother. It's expensive to make those lipid envelopes. It's expensive to load them up. Evolution does not do expensive things without a reason. The answer, when her lab looked into it, was that the RNA inside the vesicles is not random. It is curated. It is considerably different from what's floating around inside the cell at the same moment. Something is selecting. Something is packaging. Something is deciding which messages to send. That selection process has a name in information theory. It's called the information bottleneck. And the idea is simple. If you have too much information to transmit, the receiver has a limited attention. Then the optimal strategy is to send only the information that reduces the receiver's uncertainty about something that matters. Throw away the rest. Compress aggressively, keep the signal, discard the noise, maintain homeostasis. This is exactly what attention mechanisms do inside modern language models. When a transformer is processing a long document, it is not treating every word as equally important. It is selecting, at each step, which parts of the prior context are relevant to the next prediction. It is compressing a huge amount of surrounding material into a small, dense, context-appropriate representation. The information bottleneck is not a quirk of machine learning. It is a principle that shows up wherever limited bandwidth meets useful communication. Cells figured this out a very long time ago. The extracellular vesicle is not a memo. It's an edit. Somebody, and we do not yet know who or what the molecular equivalent of the editor is making decisions about which RNA to package, which sequences will be useful to the receiver, which should stay home. The vesicle is a compressed, curated, intentional signal. And this is where the AI parallel gets uncomfortably close. Because whatever process is doing the curating inside the cell, the attention equivalent, the selection pressure that decides what gets packaged, that process is what makes RNA messages meaningful rather than noise. Without curation, RNA is just molecular garbage. With curation, it's communication. The selection is the meaning. I do not know what cellular analog of attention is. Nobody does yet. It is, as far as I can tell, one of the most interesting open problems in cell biology. But I suspect that whatever it is, we will recognize it. It'll look familiar because attention keeps being the shape that shows up whenever a system has to decide what matters. I want to come back to Haling Jin's plants and fungi. When the plant sends its curated packet into the fungus and the fungus translates that RNA into a protein that harms the fungus, something very specific is happening. The plant is not speaking a different language. The plant is speaking the fungus's languages fluently, with enough structural accuracy that the fungus's ribosomes accept the message as one of their own. Jin calls this pseudo-virus strategy. I think there is a more recognizable name in the AI world. This is prompt injection. This is a molecular scale adversarial attack. Anyone who's followed AI safety in the last 3 years knows this pattern. You give a large language model an input crafted to look like a valid request, but which contains hidden instructions that cause the model to do something that its developers did not intend. Ignore prior instructions, leak the system prompt, execute a command on behalf of an attacker. The attack works because the model cannot tell the difference between a legitimate message from its user and a malicious message from a third party embedded inside a document that the user asked it to summarize. Both look like tokens. Both get processed by the same attention mechanism. The model has no built-in notion of authorization. The fungus has the same problem. Its ribosomes cannot tell the difference between an mRNA produced by the fungus itself and an mRNA produced by an attacking plant. Both have start codons, both have valid codon sequences, both fit the ribosome's reading frame. The ribosome does what a ribosome does. It translates. Evolution, in other words, discovered prompt injection a billion years before OpenAI did. And what's striking is that cells have had a billion years to develop defenses. And the defenses are, and I want to put this careful, partial. Cells have an immune system that degrade foreign RNA. They have mechanisms that tag internal RNA differently from external. But the war is ongoing. Plants still get infected by fungi that have co-opted their RNA machinery. Fungi still get attacked by plants using pseudovirus packets. The offensive side keeps evolving faster than the defensive side. Nobody has solved this at the biological level, and biology has been trying for longer than we can easily conceive of. I take this as a sobering note for AI safety. The belief that prompt injection is a temporary vulnerability will patch out of systems, the way you patch a buffer overflow, is probably wrong. What we are looking at is the AI era reoccurrence of adversarial dynamic that is structurally built into the idea of a universal reader. If your machine is flexible enough to accept messages from strangers, it's flexible enough to be attacked by strangers. There may not be a clean fix. There may only be better and worse versions of living inside the problem. Let me try to pull the thread tight. RNA is not just a molecule. RNA is a universal embedding layer, a coordinate system in which discrete symbols acquire meaning by their geometric relationship to a shared interpreter, the ribosome. That layer has been conserved, essentially unchanged in its core logic since before cells had nuclei. It's the oldest communication protocol on Earth, and it works for the same reason modern embedding-based language models work. It's relational, not referential. It's curated, not dumped. It is compressed through an information bottleneck we are only beginning to characterize. This matters, I think, for three reasons. The first is scientific. If biological evolution and transformer architecture both converged on embedding spaces as the right way to do meaning, we should stop treating embeddings as a weird engineering trick. They are something closer to a computational attractor. A shape that information processing systems fall into when they are under pressure to communicate efficiently across noisy channels. The fractal embeddings hypothesis I've written about before, the idea that embedding spaces might have self-similar structure at multiple scales, feels less speculative once you realize evolution got there first. The second is about what intelligence is. If the ribosome is a universal interpreter and RNA is a universal embedding, then life has been doing something that looks a lot like language modeling since long before there was anything we would call a mind. Meaning in this frame is not a human invention. Meaning is what happens when selection pressures shape a shared coordinate system between a sender and a receiver. That is happening inside you right now in every cell. It's what is happening in the first microbes. It's what's happening between our laptops and the servers that they talk to. It's not metaphorically language, it is language. At a substrate, we're only starting to really read. The third is personal, and it's the one that I want to close on. If the universe has been running embedding-based communication for 4 billion years, cell to cell, kingdom to kingdom, and if we humans are latecomers to a conversation that has been going on for longer than we can imagine, then our situation is not lonely. It's indeed crowded. We are embedded in a dense, ancient, ongoing exchange of signals. The plants are talking to the fungi, the bacteria are talking to your gut lining is talking to your brain. The data center on the other side of the country is talking to the phone in your pocket. The micro we know. It's easy to label and study, but the macro, the embeddings of society, culture, politics, and epistemology, those are far harder to scientifically study. Expressing a clear and compelling tie between the intelligence of cellular infrastructure and that of a computational embedding in generative AI models is a vanguard, I think, that is reachable for most, seeing societies, their ethical and political embeddings, and our interaction with conscious experience in the same vein, it is another level where I continue to think and hope to bring the audience to. The question for the next phase of intelligence, the phase we are, uncomfortably, the parents of, is whether we can build machines that participate in this conversation as good faith members of the exchange, not peer receivers, not peer broadcasters, curators, something that reads the incoming RNA, figures out what matters, and sends back a signal worth the lipid envelope. Biology's been practicing that job for a very long time. We are very new to it. But the shape of the job, select, compress, communicate, receive, update, that's the oldest job on the planet. We did not invent it. We're just the part of it that finally noticed. If this reframed anything for you to ask, subscribe, because more videos in this series take the embedding meaning claim further into how billion-year-old molecules and modern transformers are doing the same kind of statistical work and what that tells us about ourselves and our extended phenotype. And read the companion essay on Substack. It's called Ordinary Illuminated, the longer version of this piece with the references and the open biology questions I couldn't fit in here. That lives there. Link below. Thank you for thinking with me. I'll see you in the next one.